It's my great pleasure to introduce our second speaker, Elizabeth Pollina, who started her new lab, actually independent lab at Washington University School of Medicine earlier this year. Liz obtained her PhD degree at Stanford University with mRNA, focusing on the chromatin as well as transcriptional regulation of cell identity and aging, in particularly the role of H3K4 trimethylation. She then joined Mike Greenberg's lab at Harvard Medical School for her postdoc training, where she's focusing on DNA repair in neurons. She has won a lot of awards, which is really hard for me to cover in this short amount of time. But I wanted to mention some like NIH Pathway to Independent Award, commonly known as K99, or Broad Institute's Next Generation in Biomedicine. And I noticed in her resume, she has mentored a lot of people during her postdoc, like 7 people on top of 26 oral presentations at different institutes and conferences. So I have she can handle all this together. But I have heard a lot of great stuff about her as a colleague as well as mentor on top of the cool science she's going to share with us today. So she's hiring at all levels, including postdocs as well as technicians. So please make sure you reach out to her directly or through us if you're interested in neuroscience on the gene regulation. So I'm really excited to hear what she's going to share with us today, that it's entitled Activity-Dependent DNA Repair in Neuronal Plasticity. The floor is yours, Liz.
Great. Well, thank you so much for the kind introduction and for the ability to speak today. As Skye mentioned, I just started my lab about 3 months ago at WashU. So today I'm primarily going to talk about the work I did as a postdoc from Mike Greenberg's lab on activity-dependent DNA repair and neuronal plasticity. So one of the most striking features of the brain is its ability to continually adapt to environmental cues throughout our lifetime. For most organisms, the vast majority of neurons that are generated at birth survive all the way up until death, while retaining a high degree of cellular plasticity that facilitates aspects of learning, memory, and behavior. Indeed, as we engage in a variety of different behaviors, these experiences will trigger changes in neuronal activity patterns in the brain that can have really long-lasting effects on the morphology, connectivity, and physiology of neurons. And this remarkable process really unfolds across long timescales, such that by the end of our lifetime, each neuron in the brain has really been shaped by its cumulative and individualized history of neural activity. So how does activity affect long-lasting properties of neurons? Work from many labs over the years, including my postdoctoral advisor, Mike Greenberg, as well as Paul Worley, Alina Devi, and other labs, have demonstrated that a substantial portion of the longer lasting effects of activity are actually mediated by de novo gene transcription that's triggered by elevated neuronal activity. So these activity-dependent programs are initiated when you have sufficient neurotransmitter release at a synapse, causing an action potential to propagate back to the cell soma, leading to membrane depolarization and an influx of calcium into the neuron. And this calcium will stimulate a variety of signaling cascades that result in the expression of many thousands of genes, and the products of which go on to really dynamically modify important processes in the brain. For example, affecting synaptic pruning, dendritic growth and maturation, the recruitment of inhibitory neurotransmission in circuits, and communication between neuronal and non-neuronal cells. So while neuronal activity is thought to be important for aspects of plasticity in the brain, it actually poses a unique threat to genome integrity in long-lived postmitotic neurons, particularly when we consider that a given neuron in the brain might undergo these cycles of activity-dependent transcription hundreds of thousands, if not millions of times across the lifetime of an organism. So what might seem surprising at first, at least it was to me when I was first learning about this field, is that many stimulus-induced genes, including activity-dependent genes, have been linked to DNA damage and gene regulatory elements during the process of transcription. So at these regulatory elements, it's thought that DNA is cut, unwound, and then resealed in a dynamic process that has the potential to create more permanent damage if it goes awry. Now, the coupling of inducible transcription to DNA damage is not unique to a neuron and has been observed across a variety of non-neuronal cell types, for example, by the work of Stu Calderwood and Mike Rosenfeld, as well as in the brain by the work of Leonard Nuke and Liwei Sa. Well, while this process is not unique to neurons, I think it really poses an enhanced risk for neurons, which cannot rely on more error-free replication-dependent repair pathways to fix any issues. And because these highly specialized cells are not easily regenerated from stem cell pools in the brain. So I think it really raises a central question. How do activated neurons protect their genomes over time? How does one balance the benefits of neuronal activity for plasticity in the brain with this risk that it can pose to the genetic code? And as a postdoc, I was really intrigued by the idea that neurons may have evolved unique protective mechanisms at play during these periods of heightened synaptic activity. And so I decided to really study factors that would be uniquely expressed in activated neurons in the brain and not present in most other cell types in the body. And there was one factor that really emerged as an intriguing candidate for future study. And this is the immediate early gene transcription factor NPAS4, which has a number of intriguing characteristics. The first characteristic is that it was shown in 2008 by Yingxi Lin and Mike Greenberg's lab that NPAS4 is really selectively translated in response to elevated activity-driven calcium signaling. So you can see here, this is a Western blot of cultured neurons that have been exposed to a variety of different stimuli. And if you look at the classical immediate early gene transcription factor in the brain, CFOS, you can see it's turned on by a variety of different stimuli. In contrast, NPAS4 is much more selectively induced by membrane depolarization-induced calcium signaling, which in cultured neurons can be mimicked by the addition of KCl to these cultures. The second, NPAS4 is highly neuronal specific in the central nervous system. So I'm showing you here, this is a t-SNE plot of cells isolated from the visual cortex of mice that have been exposed to light. If we look at the expression pattern of a gene like CFOS, it's found in both neuronal and non-neuronal clusters. In contrast, NPAS4 expressing cells are much more restricted to neuronal clusters. So this is a activity-specific pan-neuronal transcription factor in the brain. And finally, I was intrigued to know more about this factor by an observation that I made early on in the lab, which was when NPAS4 knockout mice develop normally until about 3 months of age, but then they show a pretty striking decrease in organismal longevity. And so how is this neural-specific activity-dependent factor influencing longevity of the organisms? Could there be new unappreciated functions for NPAS4? To address this, I decided to take a classical biochemical approach, reasoning that if we could really purify NPAS4-containing protein complexes directly from the brain, we could identify new cofactors that expand NPAS4's biochemical activities and therefore its functions in the cell. So to do this, the first question I asked was, does NPAS4 actually integrate into a stable multi-subunit protein complex in the brain? To address this, I generated hippocampal lysates from animals that have received increased stimulation and separated these proteins according to size and charge using size exclusion chromatography. We then ran these fractions on a non-denaturing gel, which will retain proteins in their native conformation and preserve any protein-protein interactions. And we were intrigued to see that a portion of NPAS4 did in fact migrate in a high molecular weight complex of approximate size 1.2 megadalton. And this is far larger than the size of NPAS4 with either of its obligate heterodimer partners. This was the first indication that NPAS4 might actually integrate into a stable multi-subunit protein complex. So when I got these results, I was very excited and I immediately tried to purify this complex and identify its components. I used an endogenous antibody, or an antibody that was raised against the endogenous protein, and followed this up with mass spec. And over again, when we did these experiments, we only discovered NPAS4 and its known heterodimer partners, which only have a molecular weight of about 0.17 megadalton, suggesting we were missing the vast majority of the components of this putative complex. We decided to take a different tactic and instead generated a tagged knock-in mouse line where we could append flag and HA epitope tags onto the C terminus of NPAS4 and its endogenous locus. And what this tool allows us to do is to really efficiently purify NPAS4 complexes directly from the brain, but under the endogenous regulatory condition. We performed anti-flag immunoprecipitations from this new mouse line coupled by mass spectrometry. And this time, we're actually really intrigued to see that in addition to the known interactors of NPAS4, we identified a number of other significantly interacting proteins. And actually, if we looked into the identity of these proteins, they all mapped to a single multi-subunit chromatin modifier known as the NUA4 or TIP60 complex. And actually, if you add up the molecular weights of all these significant interactors, they equal just about 1.2 megadalton in size, suggesting to us that we had identified the majority of the components of this putative MPAS4 complex. In addition, we found that like MPAS4, many components of this complex are highly enriched in their expression pattern in neurons in the brain, indicating that this complex may have a specific biology. So in response to neuronal activity, we have identified this protein complex that assembles between NUA4 and this MPAS4 activity-dependent transcription. We really wanted to understand what could be the functional significance of this. And one obvious prediction is that this complex is important for modulating chromatin and therefore activity-dependent transcriptional responses. However, I was also intrigued by work in yeast, flies, and non-neuronal cells, which has shown that a conserved and important function of the NUA4 complex is actually in stimulating DNA damage repair, particularly the repair of DNA double-stranded breaks. And I thought this was really interesting in light of the observation that many activity-dependent genes seem to acquire signatures of DNA damage upon neuronal stimulation. And so we wondered, could the coupling of an activity-specific transcription factor like MPAS4 with the chromatin modifying capacities of NUA4 be a way that neurons can efficiently couple transcription directly to DNA repair in order to preserve genome fidelity at these plasticity-associated genes over time? So actually the remainder of the talk is going to be our exploration of this dual function of the MPAS4 and NUA4 complex in both transcriptional activation and DNA damage control. So in order to begin to dissect what this complex is doing, we needed a system where we could perturb different components of the complex and look for their effects on both transcription and DNA damage repair. And so we developed the following experimental paradigm. We can take MPAS4 floxed or NUA4 component TIP60 floxed mice and inject one hemisphere of the hippocampus with a virus expressing CRE tagged with M-cherry. And on the contralateral side of the brain, we inject a nuclease deficient version of CRE, which cannot excise the gene. And what this allows us to do is then within the same animal to compare cells that have intact MPAS4 and NUA4 signaling to those that do not. It helps to control for animal-to-animal variation in the activity state in the brain. So we then use this system to first ask, how does perturbation of this complex influence activity-dependent transcriptional responses to neuronal activity? We decided to use a single-cell gene expression approach where we could really look at transcriptional programs across a variety of different neuronal cell types. So in these experiments, we can isolate nuclei from MPAS4 floxed or TIP60 floxed mice injected with CRE and therefore conditional knockout cells, or delta CRE and therefore wild type cells. We performed a standard 10x genomics experiment, but added in one important modification where we used a nested PCR to amplify viral transcripts, allowing us to very confidently assign each of these cells as either a CRE or delta CRE infected cell. As you can see, this is the results from our MPAS4 flox dataset. We were able to capture the major cell classes of the hippocampus. And using a bioinformatic pipeline, we were able to assign each of these cells as either conditional knockout or wild. And what we observed was that within a cell class, for example, if we look here at these CA1 neurons, you can see there's a separation between conditional knockout and wild type cells, despite not using any information about viral transcripts and clustering. And this indicated to us that there really were substantial changes in gene expression when you remove components of this complex. We use this data to ask whether MPAS4 and NUA4 complex component TIP60 would actually coordinately regulate gene transcription. So we had demonstrated that they do in fact associate biochemically, but do they work together functionally? To address this, we defined MPAS4 target genes as genes that were significantly downregulated when you lose this MPAS4 subunit of the complex. And what we found was that across each of the distinct neural classes in the hippocampus, downregulation of or knockout of TIP60 caused a downregulation of MPAS4 target genes. The converse was also true. If you define genes significantly downregulated upon loss of TIP60, they are also coordinately downregulated upon loss of MPAS4, demonstrating to us that these two subunits really do coordinately regulate gene expression programs in the brain. Moreover, when we looked into the identity of these different programs, they were in fact distinct across different types of neurons, indicating that one important function of this complex is to specify cell type specific transcriptional responses to neuronal. We then wondered, would perturbation of this complex have an effect at the circuit level on activity-dependent changes in neural circuitry? We wanted to define an electrophysiological phenotype for this complex, and also to further prove to ourselves that MPAS4 and NUA4 complex components would phenocopy each other and therefore really were part of a functional complex. We therefore turned to an electrophysiological assay we use frequently in them. So one important modification to neural circuits that occur in activated excitatory neurons is to actually stimulate the levels of inhibition that are recruited onto those excitatory neurons as a way of maintaining balanced excitation and inhibition in activated neural circuits. And we wondered whether perturbing components of this complex would affect this activity-dependent recruitment of inhibition. So in these experiments, we can sparsely inject MPAS4 TIP60 floxed mice with our virus-expressing Cree tagged with cherry, and then in SBLISS perform paired simultaneous recordings of evoked inhibitory postsynaptic current from a wild-type cell or a neighboring conditional cell. As we had seen before, when you remove MPAS4, you have a decrease in the activity-dependent recruitment of inhibition, only in the context in which animals have been stimulated 24 hours prior and have therefore had time to turn on activity-dependent genes. What was quite exciting to us in these experiments is that when we performed the exact same experiment in our TIP60 flox mice, we saw a precise phenocopy, suggesting these complex components do in fact work together, and that one important function of this complex is to stimulate the activity-dependent recruitment of inhibition. So just to summarize what I've shown you so far, we have biochemically identified an interaction between MPAS4 and the NUA4 complex. And this complex has 21 subunits and assembles across multiple brain regions. By single-cell RNA sequencing, we found that MPAS4 and NUA4 actually co-regulate distinct gene programs in different types of neurons in response to elevated neural activity, and that this complex really functions together at the circuit level to promote neuronal inhibition. So in response to neuronal activity, this complex assembles, and one critical function is to stimulate activity-dependent transcriptional programs. But what about this second proposed function? Complex? Does it actually stimulate DNA damage repair at its target sites? In order to address this, we needed to identify where this complex binds in the brain and whether sites bound by this complex are undergoing damage in response to elevated neuronal activity. To do this, we adapted cut and run to look at the binding of several components of this MPAS4 NUA4 complex in freshly isolated hippocampal nuclei. I'm showing you here sequencing traces from our cut and run assays for several components of this complex. And you can see that they co-localize at the promoter of this new target gene, RGS7BP. We then asked, does this gene associate with signatures of DNA damage? Looking first for a well-known chromatin signature of damage, phosphorylation of the histone protein H2AX, which is known to occur at the vicinity of a double-stranded break, although it can also spread many kilobases away. And what we observed was that there was in fact an increase in this gamma H2AX signal when we stimulated the neurons. This was true not just at this example locus, but in fact true across all activity-inducible genes that are bound by MPAS4, as well as the regulatory elements themselves, the enhancers and the promoters, with the highest levels of gamma H2AX induction occurring at the sites that had the most MPAS4. So this indicated to us that the complex was binding to sites that had chromatin-based signatures of DNA damage. But we wanted to further corroborate this by trying to more precisely map locations of these DNA double-stranded breaks in the brain. So we adapted a protocol called BLISS sequencing, which allows you to get better base pair resolution on where breaks are occurring. We took animals across a time course of stimulation, isolated nuclei, gently fixed them, and then in situ ligated sequencing adapters onto the free ends formed from a DNA break that contain unique molecular barcodes. And what this allows us to do is then to make libraries that allow us to map the location of a single DNA double-stranded break in a single template molecule. As you can see here from the sequencing traces of this assay, we're able to more precisely locate the DNA break to the promoter of this gene, RGS7BP. So what happens now if we look at all activity-induced regulatory elements, or elements that increase transcriptional activity in response to neuronal stimulation? We found that there was, in fact, an increase in DNA double-stranded break signaling at 2 hours post-stimulation of these neurons. And intriguingly, that there was the maximal induction at sites that had the highest levels of MPAS4. And this was interesting to us. It suggested that MPAS4 might be specifically targeting sites that are undergoing high levels of damage. And in fact, we compared using 2 independent DNA break mapping techniques, one sBLISS-seq, one called nSeq, and found that sites that were bound by MPAS4 had higher levels of DNA breaks at 2 hours post-stimulation, and sites bound by the other activity-dependent transcription factor people often study, CFOS. And this again indicated that this complex might be importantly targeting sites that are undergoing activity-induced DNA damage. But do these sites also undergo repair? We decided to look at this by examining a time point that was far after we'd stimulated the animal, so 10 hours post-stimulation, when activity-dependent transcription itself is beginning to return back down towards baseline. So as I showed you before, stimulation of the neurons increases DNA break signal, and at 2 hours post-stimulation, we began to see a resolution of this DNA break signal back down towards baseline. And this suggested to us that the complex was binding to sites of both DNA damage and repair in activated neurons in the brain. But does the complex actually stimulate the repair process? In order to address this, we wanted to again perturb the components of this complex and look across a time course of stimulation in the brain to DNA double-strand break signals. And what we observed was that loss of MPAS4 causes an increase in DNA double-stranded break signal, particularly at late time points when many wild-type cells have begun to resolve those breaks back down towards baseline. This suggested to us that the complex could, in fact, be important for helping to stimulate repair at its target sites. So how might the complex actually stimulate repair? One idea is that by modulating chromatin, the complex could help to recruit and retain additional DNA repair proteins to the genome. So we decided to look for a well-known sensor of DNA double-stranded breaks, an early responder to these sites, a complex called the MRE11 or MRN complex. We looked by cut and run for the binding of this repair complex in the brain. And we observed that 2 components of the complex really nicely co-localized with MPAS4 binding. This was true at its target gene, RGS7BP, and was also observed across all MPAS4 binding sites. So MPAS4, NUA4, does in fact co-localize with the MRN complex in the brain, but does it stimulate the recruitment? We again perturbed the system using our Cre-delta-Cre system and FACS sorted out nuclei that were either Cre expressing or delta Cre expressing and performed cut and run. And what we observed was that there was a significant reduction in the recruitment of the MRE11 subunit to MPAS4 bound sites, as you can see here again at this nice target gene RGS7BP. So it appears that the complex is in fact helping to stimulate repair in part through the recruitment and retention of additional DNA repair machinery to the target sites. Now, one prediction from this model, if in fact this complex is playing a protective role, is that over time, sites that are protected or bound by this protective complex may have a suppressed accumulation of mutations with aging. So we really wanted to test how do mutations accumulate at different types of regulatory elements in the brain, comparing transcriptionally active sites that are bound by MPAS4 to those that are not. To do this, we isolated neuronal nuclei from animals at different ages, and then performed a mutational analysis at sites that are bound by MPAS4 versus sites that had comparable levels of histone acetylation, chromatin accessibility, but were not bound by this complex. And in these experiments, we incorporate a unique molecular barcoding strategy to help us understand what's happening in a single template model. Interestingly, in the numbers of sites that we looked at, we found that sites that were not bound by MPAS4, but were transcriptionally active, showed a steady increase in mutational accumulation. In contrast, MPAS4 bound sites, despite being transcriptionally active, did not show this same accumulation, suggesting that the complex may help to protect against mutational accumulation. In addition, in preliminary data, we have been collecting neuronal nuclei from older NPAS4 mice, looking specifically for signatures of failed double-stranded break repair insertions and deletions. And what we have noticed is that there's a tendency towards increased insertions and deletions in NPAS4 knockouts, particularly at NPAS4 bound sites, which is not observed at sites not bound by the complex. So this, taken together with the data from our wild-type aging gradient, suggests that this complex may help to suppress more permanent damage in the form of mutations. Now, is there a physiological or organismal consequence to a loss of the functions of this protective complex? I just want to remind you of the strong phenotype that we observe in terms of longevity of these animals. And while we still don't understand exactly what causes this premature death, it's tempting to speculate that it arises from multiple critical functions of this complex gone awry, including dysregulated gene expression, decreased inhibition, and accumulating DNA breaks and mutations. And certainly it pinpoints this NPAS4 NUA4 complex as really critical for potentially long-term health and survival of the neurons. So with that, I'll just conclude by saying we have identified one particular mechanism that neurons employ to both rapidly activate transcriptional responses that can promote plasticity at the circuit level, while also helping to stimulate repair of these sites to prevent the accumulation of more permanent damage over time. And we're intrigued by the idea that this complex may be very important for helping neurons to sustain the appropriate responses to transcriptional stimuli throughout their long postmitotic lifespans. We're very interested in the lab in trying to understand whether damage to activity-dependent regulatory elements could be a significant cause of cellular dysfunction in both aging as well as certain forms of neurodegeneration. And in fact, this work has really inspired the bigger picture questions that we're interested in pursuing, namely really trying to understand how the nervous system constantly adapts to environment, particularly at the level of transcription and chromatin over the lifespan of an animal, how these pathways are different in species of vastly different lifespans, and how specific interactions with the environment can impinge on this process. How does diet, exercise, sleep pattern really change neuronal activity patterns, as well as changes to chromatin and DNA damage? Specifically, we are really interested in understanding more about how different types of neurons in the brain, all of which have very different transcriptional levels, as well as levels of neuronal activity, how they can protect regulatory elements from damage over time. So we're modulating neuronal activity levels in NPAS4 complex knockouts, looking at changes in damage as well as transcriptional programs. We're also coupling this with an examination of longevity and behavior. We're interested in expanding this to look in humans who have to survive much, much longer, using both postmortem human brain tissue, as well as cultured systems where we can screen for factors that could promote repair and activate it. And finally, as I mentioned, we're really interested in studying how different environmental stimuli can impinge on this process, with a focus on how changing patterns of sleep influence damage and repair in the nervous system. And so with that, I'll just conclude by thanking my co-author on the work I've presented, Daniel Gilliam, who was really a wonderful collaborator for all the studies I've presented, my postdoctoral mentor, Mike Greenberg, wonderful collaborators at Harvard, especially Andrew Lando from Bernardo Sabatini's lab. And thank you so much for your time and attention.
Great, thanks, Liz, for a wonderful presentation. That's a lot of work. And so if you have any questions, feel free to type it in the Q&A box or raise your hand. So we have 3 raised hands already. I'm going to unmute Ashishi, sorry. Okay, maybe I will unmute Jason first since
Hello?
Yeah, we can hear you.
Okay, good. I wasn't sure if the first one didn't work. Yeah, really cool talk. I was wondering, it's really interesting because in NPAS4, I guess is some kind of transcription factor. Is it is it recognizing some kind of motif, and that sequence motif tend to generate double-stranded break in the neurons, or it's binding to some generic, how is it attracted to the DNA damage signal, or can it just bind generic double-stranded break or something?
Yeah, I mean, that's a really good question. We don't really know why it is that it seems to be enriched at sites where there's more DNA breaks. What we find is that it seems to be binding with a lot of GC-rich, close to GC-rich motifs, which I suspect probably contribute to why we see more damage there. It also binds more often to promoters than a factor like FOS, and we know that at least in neurons, the activity-dependent damage seems to be more localized to these promoter regions.
Oh, interesting. Great, thank you.
All right, Yuki, Yuki, I'm going to unmute you now. Yeah, you can speak now.
Can you hear me now?
Yeah.
Okay, thank you for a wonderful talk. Actually, I have several questions. And first, so at your first part, you talked that MPAS4 knockout will result in the shorter lifespan. So I was wondering whether you can see a kind of neuronal death because of the DNA damage that is accumulating neurons. And the second question is about what kind of mechanism are you considering to repair DSP? So I think that mRNA complex is functioning in the early phase of DSP repairing systems. And after that, several proteins related to maybe non-homologous engineering, but also maybe homologous recombination would happen. So have you ever find any kind of key proteins that would function after mRNA and complex accumulation? And okay, I'll stop here.
Yes. I'll answer the first two. One is, interestingly, we don't actually see, we have looked specifically for apoptosis in the MPAS4 knockouts and haven't seen anything super obvious. My suspicion is that, but we also haven't done that comprehensively, and we looked a little bit earlier probably than I would like. We could look a little bit later. So there's not massive cell death there. And really why it is that they don't survive, I think is an interesting question that we kind of want to
exactly.
More specifically. If I had to guess, I think it's an accumulation of essentially subtle mutations and then potentially dysregulated inhibitory control in the brain that could be causing subtle seizures, but we haven't really been able to quantify that. These animals could potentially also have a circadian defect, which also could contribute to sort of some of the organismal phenotypes that we have. As far as what's happening there, I'm really interested in this question. So work from yeast and non-neuronal cells would suggest that in replicating cells, one of the major ways that TIP60 promotes repair is through the suppression of NHEJ and promoting homologous recombination. Does that work in the brain, and how so? We don't actually really know. What we're trying to do is figure out, actually, is there any end resection going on at these sites in neurons? And if there is end resection and a suppression of, let's say, 53BP1 binding, which would be part of NHEJ, if that's true, then what is the template that's serving as a more homology-directed repair? And there's some interest in the idea that potentially RNA could be filling that in. And so those are things we're just interested in seeing. We suspect that some of this is mediated by NHEJ, but it is also intriguing to explore the option that maybe the complex is helping to kind of shunt to a less error-prone pathway in neurons. I would argue that we, at least in the nervous system, are still kind of in the infancy of understanding how all of these repair pathways work. We know a lot about what's going on in dividing cells, but I think particularly in intact circuits in the brain, we still don't quite know how neurons resolve these kinds of things.
Thank you very much.
So you also have a question in the Q&A box. Also wondering more about the yeast MPAS4 complex, whether there are something in the yeast that would be useful for you.
Yeah, that's a great question. I mean, a lot of the work, a lot of the concepts were inspired from the work in yeast. And the reason we sort of started thinking about DNA damage was because of what had been discovered in yeast. In addition to thinking about using the mRNA complex to kind of, is some evidence that there could be interactions between MRE11 complex and NUA4 from yeast. So yes, we are always sort of looking in the literature from other organisms to inspire sort of the ideas that we're taking into the brain. But as I mentioned, I think At least I'm keeping an open mind because I don't know how these processes will play out in a postmitotic mature neuron.
That's great. Yes. Thank you for the wonderful presentation. I think we can end here. But again, Lisa is recruiting postdoc and technicians. So if you are interested, please make sure you reach out to her. And let's thank both speakers for the wonderful presentation today. Yeah. Thank you both.